Hey guys, back at it with biology and AS Biology in a single month, day 9, and this is the second paper of the paper 2 series. So yeah, this is the 2017 paper, and yeah, like in the previous video, I did the specimen paper, which actually I did horribly on. I got about 33%, but in this one, you get some improvements. So we're up 66%, which is very good. You can see I'm just learning about, I think that was, actually can't remember, I think that might have been, oh yeah, the root tip squash experiment. Just seeing the mitosis in action, and yeah, I was also able to complete this paper a bit faster, so this video is 4 minutes again, but it's only been sped up times 30, so that's really about 120 minutes, or just about 2 hours, which is pretty good. I'm hoping to get that down to about an hour and a half as we do more and more papers. Here you can see a tensile strength, which was relatively easy because I do physics. And oh yeah, so yeah, this is about the shortening of the fibers. I didn't realize that the fibers shortened during metaphase is what caused it to pull apart, but now I do. Also had a bit of a weird theory with this one. Turns out there isn't any correlation between the diameter and the tensile strength, although it did seem like that from the data. But it's alright. Again, another stem cell question. Right, if noticed if you watched the previous video that there was a similar question. This one just put into context, but it's the same idea, just the stem cells get differentiated by having genes in their DNA activated, which produces proteins which control the function of the cell. And then, again, this was just a bit of a exam technique sort of error. I also believe there's a lot of clinical testing in topic 3, so topic 3 is a bit confusing. It seems to have all of the leftover topics, which is a lot, but that's alright. I had to learn about William Wiffering and his testing. Basically just used some patients of his to test his new digitalis soup, and I guess if it didn't work they just die. Yeah, this is all fine. Eukaryotic and prokaryotic. So yeah, this was an extension to GCSE. There are 5 points here. I had to get all of them, and then I spent a lot of time on these conservation questions for some reason, but that's fine. Moving into the last 20% now, so this is the bit which usually takes the longest. It'll be interesting to see like a curve of how quickly the first bit goes, but I guess it would just be something like any other negative exponential curve. Sorry, not negative exponential, yeah, negative exponential, and then translated up by 1, that's what I meant. Biodiversity, this is again renewability of bioplastics. The juice, I learned from my mistakes and didn't use a diameter at all. Tissue structures, yep, that's fine. Just redoing the questions to get 100% of the marks, which lets me just get a better idea of everything to do with it. Into these last stages, 93. So yeah, it's actually crazy that the last 30 seconds is all just the last 7%. That means it took about 900 seconds, or, okay, 10 minutes is not too bad. 10 minutes, about 15 actually, but yeah, it's not actually too bad. And about chromosomes, autosomes, no, actually still not sure whether all autosomes have the same identical DNA or if they're separate. Like each autosome has a different part of the DNA. Still need to learn about that. Hopefully it comes up in future questions. And once I do these questions, I'm going to start focusing on just extra resources to sort of consolidate knowledge. But I'm saying this is a good go. And yeah, thanks for watching. See you guys in a future tutorial. Bye.